Hello, I'm Moyun Wu.
I'm Dayo Huang.
We're presenting our paper, Expansion Microscopy Imaging, Isotropic Restoration by Unsupervised Deep Learning. Expansion microscopy combined with light sheet microscopy enables high-resolution mapping of neural connections and pathways at the organ level. However, reconstructing 3D neural morphologies remains challenges due to two factors. Firstly, the axial view, as shown in the purple window, has lower resolution than the lateral view as shown in the yellow window, due to the point spread function. Secondly, inhomogeneous fluorescence labeling and distribution of neurons, marked by the arrows, lead to visual ambiguity between neural morphologies and the background. To overcome these challenges, supervised deep learning techniques have widely been utilized. However, they require extensive manual annotation, which is laborious. Therefore, we propose an unsupervised model using multi-scale deconvolution and gain-based domain adaptation. We first train a single-scale deconvolution model to map synthetic microscopy images to their corresponding neural structures. Synthetic images were generated using theoretical PSS, Poisson and Gaussian noise to emulate DPM neural structures. The original images were resampled into different physical scales, and multi-scale responses of the deconvolution model were weighted to create MDCs through Fourier transforms. MDCs were generated for both lateral and axial views before proceeding to the next step. Next, we employed unsupervised domain adaptation techniques using gain models with cyclic consistency. Our gain models consisted of two generators and two discriminators to translate and ensure visual similarity between different views of images and MDCs. During training, we combined the MDCs and original images as input and simultaneously optimized them. This approach enabled our model to capture the spatial structures of neurons at different physical levels.
Okay, let's see the result. In the first row, we have the original image, and in the last row is our isogain result. Now, if we look at these three rows of image, we can clearly see that the imagery enhanced by the isogain shows some improvement in neural continuity and imaging resolution. Now, let's focus on the neural continuity. Let's see the yellow arrow. It indicates that isogain is capable of enhancing the connectivity of those faint neural sections. Now, let's move on the imagery resolution. Here, we can see that neural can be distinguished from the background signal by using isogain. So, this result demonstrated that using isogain to enhance imagery leads to a significant improvement in the quantitative measurements of the reconstructed morphology. Then, we also see the reconstructed imagery can also power to quantitative measurements between in neural and protein. Finally, we reduce the SL digital resolution and using isogain to reconstruct it. In this figure, we successfully reconstructed detailed 3D neural morphology. As a result, our isogain enhanced SL resolution, approaching isotropic resolution in expansion microscopy imaging. This breakthrough has significant implications for understanding neural structures. Thanks for your listening.